Now we have two more lectures before the lectures of Dr. Mia, but now we have a postdoc lecture, Wafa Ali, from the Laboratory of Paleontology. Your supervisor is Professor Max Lager. Okay? Wafa? Thank you very much. Wafa is the unique postdoc presenting in the symposium, I think? Yes.
I'm going to go to this side, the other side. So, good morning everyone. My name is Wafa Al-Halabi, as you pronounce it, but the real name is Wafa Al-Halabi, which is a little bit different. Okay. First of all, thank you a lot, Professor Melton, to invite me to this presentation. It's always a pleasure to talk about my experience in Brazil. In contrary of all of you, I'm telling my experience in this country, not outside this country, which is my long internship. It's almost, I'm entering my 8th year in this country. I chose a topic, it's don't think outside the box, think like there's no box. The life-changing story of a Syrian woman in Brazil, because it is a life-changing story for me. People, they date their life like before and after. I'm dating my life before and after Brazil, so it makes a huge difference. So I'm going to start with some important dates in my life. In 2004, I graduated from the Biological Science faculty in Damascus University. And then in 2007, I did my master's degree in environmental science. Nothing related to paleontology back then, because we don't study paleontology. I don't know why. Maybe probably because we have some those religious restrictions about evolution and those stuff, so we really don't study paleontology. After that, I started to work as environmental consultant from 2008 to 2016. A lot of environmental work, a lot of field works and with industrial systems, a lot of men, and working with men is not very easy, so you don't get offended. Sometimes in some countries, like those photos are in Iraq and Libya, where I worked, as a woman, I had to wear a head cover because it's not acceptable for me to work on those parts of the world without using some religious restrictions. During this time, from 2008 to 2016, I got introduced in 2009 to the concept of paleontology. Like, wow, there is something that's called paleontology, and I was introduced to it by several archaeologists that I work with them. I would like to honor one of them, Mr. Khalid Asad, who was a big archaeologist in Syria, and he was beheaded by ISIS in Syria trying to protect the archaeological sites in Palmyra. So in 2010, I started to run after paleontology. I wanted to do paleontology, so I started to, first of all, my passion was fossilized pollens, insects in amber and all those stuff. So I collected the material from Syria, and we could identify the first fly from Syria. The species named Blodani, it's from the city where I collected the amber. Then the journey started. I want to do paleontology, so I started to run after paleontology. Well, but the war started in 2011, so it was like impossible to continue, and I have to switch on my survival mood to work, to help the family, to travel around and all those stuff. So all plans just like stopped. I couldn't do anything. Until 2016, I decided to come to Brazil for so many reasons that I'm gonna list them because there are a lot. And I started to look for a paleontologist. I said, okay, I'm coming anyway to Brazil, let me do paleontology, that's what I always wanted to do. And I found Professor Max Langer, and it was, I do believe in hard work, but sometimes luck is very important in life. I was so lucky to get Max as my professor. He accepted me directly, and he helped me a lot. He gave me finance when I first started to work as a fossil preparator, and we developed the PhD project together. So it was like a great help and amazing welcoming. Because of this help, I changed my course. I wanted to do insects in amber or pollens. So I said, no, I'm going to stay in this vertebrate lab because the warmth, the love that I got from the paleontology lab here in Departamento. So we developed our first project, something close to what I wanted, to study vertebrate micro-remains. And I worked on this project for 7 months. Then, a British paleontologist, Matthew Byron, he published a new hypothesis about the relation, the phylogeny of the early dinosaurs. In dinosaurs' world, you have the two main lineages, the Saurischians and the Ornithischians. And it's a very traditional, very well supported theory. Then he came and he revived an old concept, and he put together Ornithischians and Theropods in one lineage and removed Sauronorth to another lineage in a clade called Ornithilidae. So the word of the dinosaur paleontologists were like collapsed. Oh my God, this is not acceptable. And Max made a reply in the same year with some co-authors to prove that this is not correct. The point is that on the global dinosaur paleontologists, it was a huge effect, but also it has an effect on me, because Max decided, Wafa, let me change your PhD project. I was like, dinosaurs, I've never thought to work with their majesties. They are so big for me to work with them. So I said, okay, let me see what can I do, what he suggested. So he suggested a project about reviewing the characters of the phylogenies of early dinosaurs. Early dinosaurs, usually they have this geological lifespan from the late Triassic to the early Jurassic. And we decided to review all the phylogenies that was published from 1970 to 2020, like 50 years of reviewing the characters. It was, I could only achieve to solve 3,000 characters. The whole list is like 6,500 characters. We only worked on the appendicular skeleton. And the result was big PhD thesis, 1,500 pages of work. Thankfully, people really like it, and we still didn't publish it, but it's on the plan. So, I finished my PhD, and now let's go back to my passion. I always wanted to do paleontology in Syria. Syria is not on the map for paleontological research. We are only on the map for war and disasters and all Middle East problems. But I want to talk about paleontology in Syria. So I started to dig in the past. What do we have? What are the fossils that are collected? So I went to Syria and I started looking for fossils. What are the fossils that have been published, where they are? And apparently I started to get noticed by media, because I didn't realize back then that I was the first Syrian vertebrate paleontologist in my country. We have a lot of micro paleontology experts, but vertebrate paleontologists. So I went to TV, I went to journals, and I did a lot of interviews. That was a pressure, but also was a blessing. Blessing because people started to contact me. Yeah, we have this fossil, this is an outcrop that you might look for fossils. So locals, apparently, they collected fossils, but they didn't know the importance of those specimens. So we started with the first fossil that was collected in Syria in 2010, but kept hidden for unknown reasons until 2017. And it is a big plesiosaur. Plesiosaurs, those are the marine reptiles that were flourished in the end of Cretaceous. Like some plesiosaurs, they could reach 13 meters in length. And what was found is 52 articulated vertebrae, which has turned to be something important when we started to study this. Max got excited about the idea and he funded my trip to Syria to study this specimen. Whilst we're studying this specimen, the idea came to our head, yeah, let's make a series of articles about Syria. It's something like the word in Portuguese that I do like to use, it's chamativo. So we chose the title, Recovering Lost Time in Syria, #1. So now we are talking about the plesiosaur, that the manuscript is almost ready, but the co-authors are giving me hard work. But hopefully at the end of the year we're gonna submit this manuscript. This plesiosaur, it's very important for the Arabic plate because it's not the biggest one that was collected among the others from Saudi Arabia, from Jordan, or from Palestine. It's the oldest. It is from Santonian Conatian age, which is around 89-86 million years, while all the rest are from the Maastrichtian and Campanian age, like 72 million years. So I hope that's going to give the right impact about it. During the research, I came across an article that was published about small distal tibia bone of a theropod from Syria. It was published in 1968. Sadly, I couldn't trace the fossil, the original fossil, but we could find the cast at the American Natural History Museum. So we asked the curator there to send us photos of the cast, and we have a theropod expert in the lab, one of our postdocs, Rafael Delcourt, and I sent him the photo, and Rafael went crazy. Wow. This is not possible. This is Abilisauridae, a group of derived theropods that they have only Gondwanian origin. They are in Africa, in the South America, and a little bit in India. And there is no registrations before about this animal in Asia. So we have to make a revision about it. And now we started a group of researchers to do the revision about this bone, also to revise all the dinosaurs that have been found in the Arabian plate because it's a gap in the world of dinosaurs. So this is the second article, #2. And #3, it's mosasaur, another marine reptile also from the Cretaceous. The lower jaw of mosasaur that was collected in Syria in 1938 during the French mandate in Syria, and I could reach the Natural History Museum in Paris, and the professor was very helpful, and we are describing now this fossil. So this is the third article on the list. Plus, contacting the locals, they pointed out, yeah, we have a possible pterosaur tibia bone from Syria. We have a cast of a turtle, which is, in the fossilized turtles in general, you have the bone fossilized, but not the internal of the carbus. So this is a mold inside the carbus of the turtle that's also on the plan to study it. And probably some Pleistocene mammals that was collected from archaeological sites, but we have the same problem as Brazil. People, they come and they steal our fossils, so trace the fossils is not that much easy. Forgetting about the scientific part, I'm gonna talk a little bit about serious challenges to be outside your country. First of all, cultural differences. Guys, I'm an Arabic woman. Brazil for me is just like the planet of Japan, it's the planet of Brazil. It's like a lot of cultural differences. So I needed the time to cope with this. Language, my God, it took me 2 weeks to pronounce definitivamente. It's very hard word to say, like, but I could. I'm proud of my Portuguese nowadays. For those who travel abroad, homesickness and loneliness, as Malu said, it's huge. It's something that you cannot deal with it sometimes. It's a lot of depression, a lot of missing, a lot of feeling alone. But you can find a way around it as long as you have a dream to achieve. Don't talk about financial difficulties, it's everywhere. It's not easy to finance yourself alone. as an independent person, it takes a lot of effort. And of course, the pandemic that hits everyone, it hit me, something that maybe not good to say, but it has also a good effect on me. Without the pandemic, I couldn't write 1,500 pages. It's like impossible. I was locked in my apartment and doing the work. The funny challenges, Brazil is a continent. Please don't say it's a country. It's a continent. It's a huge. I'm getting lost in Ribeirão, so imagine I have to travel to another state. It's like very big for me. I cannot understand the standing distances in this country. It's not possible for me to get it in my hand. And about something you use a lot of different hand gestures. Yeah, something that is not acceptable. We say 3, you say 3. This hurts your hand, it's not acceptable to say 3. And you have too much partying and very friendly environment. It is nice, it's very nice, but it's hard when I'm gonna decide to leave the country because I'm looking for a job, I have to work. So there's no other options. But I fell in love with this country, which is now I have 2 loves. Syria, which has always been my love, and now leaving Brazil, if I could find a job, is not really easy. I wanted to mention Brazilian people. I will never forget my first aula trote. I don't know how to say this in English. And the welcoming and the warmth that the students gave to me. It was like perfect, really perfect. And they were terrified that I was talking in Arabic with them, but still they loved me. And amazing moments in Palolab, like Palo Natal, it's our favorite occasion. Festa Junina, guys, this is the best occasion in Brazil. I love Festa Junina. Fieldworks in this country, like amazing. And recently I went to Peru, so it was like another experience. Plus a lot of conferences, a lot of places to present your work, to talk about your achievements. And one of the funny challenges in this country, guys, why you put animals like this in the butcheries? I turned vegetarian because of those stuff. It's not acceptable. Try to take care of animals. It's not very good. I want to thank everyone who helped me during this process. All the agencies, all the people that they helped, and I wanted to reflect on last 2 points. One point that Vinicius said today, Brazil is amazing. Don't get crazy about being abroad because we have good science, we have very good people, and we have a very friendly environment that you cannot find it any other place in the world. So grace that you are Brazilians. I am now officially Brazilian. I got the citizenship. So I want you to appreciate that you are Brazilians. And something else that Professor Thomas said yesterday, don't compare yourself to others. We are individuals. Each one of us has a different path in life. Me personally, I decided to quit my PhD several times during the process, but I could continue. And one last word for women in this place, because I'm a big supporter of women. If an Arabic woman could break a lot of taboos, you can conquer the world. So please, believe in yourselves and do whatever possible to be the one that you want to be. And thank you all for listening, and I'm ready for questions.
So, did you have the opportunity to do field work in Syria to find some fossils, or?
I went to visit the plesiosaur locality. Sadly, it's located in a phosphate place where they extract phosphate. and most likely they are crushing all the fossils that they get with them. But nowadays I'm working with one of the, I say this is a little bit big to say, but I'm working with one of the ministers there to issue a law about protecting fossils in Syria. Because my future plan, I want to do excavation in Syria, and I promised Professor Max, we're going to go to Syria to find fossils.
No, that's okay. You mentioned that you found some fossils from Syria in France. Is France the country that probably has the most specimens of those things from Syria, or would you find it elsewhere, like in the US or Germany?
French people, they occupied Syria for around 25 years. So they have a lot of fossils. They took everything with them. Dutch people, because with archaeology work in Syria is very big, so Dutch people, the archaeologists that they were going there, they also collected fossils. All those mammalian fossils are with them. So in Syria we have very little, and if we have, we don't know where they are, because to be honest, this plesiosaur was thrown in a basement. So like the whole new concept about vertebrate paleontology in Syria, I'm working on it. But as we don't have a museum for paleontology, for example.
No, I think I can say it without the microphone. So very inspiring, your talk. Thank you very much. I should confess that I was terrified with your aula trote. It was terrible. I said, this is university, oh my God, what am I doing here? But I would ask you, I would like to ask you about, you said you went back to Syria in the middle of the war. How was this process? What the difficulties you faced there at that moment?
Look, I lived during the war in Syria. I left Syria in 2012. The war started in 2011. The worst part about a war that you lose your humanity. You don't feel anymore for humans. I'm a big fan of cats and animals, not anymore of humans. I don't believe in humanity. Sorry to say this, but I'm a big supporter of the extinction of human beings. They don't deserve to exist. The way that they killed each other, the way that they treated each other, it's not possible and not acceptable. So the long-term effects even higher than living in the war, in a military war. Like for example, a lot of women I know, they decided not to have kids anymore. Why should I bring a kid to this world? It's not possible. So war is the worst thing ever can happen to anyone.
Thank you, Wafa. Just one more question. You said about the ISIS in the beginning of your speech. Is it safe now in Syria in some places and other places that are not safe? For example, if I want to travel to Damascus, for example, is it safe today, these days?
The point is that now we are, imagine Syria is a cake and it's getting divided by several countries. Like Russia wants its part, Iran, United States, the Britons, everyone wants its part. So Damascus and all the coastal line are okay to go. I cannot say safe and define safety. It's like, you might be kidnapped. It's possible as a foreigner to be kidnapped. But if you go very well with like armed people and protected, like I was doing my work in Iraq, I was always accompanied by armed people. You can do your job. But definitely you cannot go to the Turkish border or to the Iraqi borders. It's like impossible. Like the possibility to get killed is very, very, very high.
It's called
The Kurdish lands, yes.
So it's no safe.
Not possible at the moment. Not possible. Plus, don't forget about landmines. Natalia asked me about the fieldwork that I did. Yes, I went to the plesiosaur locality, but I was following a very small path like this because they cleaned it, but the landmines all around, so you cannot just like 1 foot wrong and you're ciao.
Thank you very much, Wafa. I think we could expend 1 hour, 2 hours, 3 hours just with this speech. And you talking your experience and about Syria and all the adventures that you had in your life. I don't know if I can call it adventure, but okay. Adventures, I don't know the correct word to it. Well, Jennifer? So this is the next PhD student's about speech with Jennifer Natalia, and she will speak about commercial pink shrimp, Pneus brasilienses. Thank you, Jennifer.
So hi, good morning everyone. My name is Jennifer Teles, and today I'm gonna present my work entitled Amazon Orinoco Plum is a barrier for the widespread and long dispersion marine pink shrimp, Pneus brasiliensis. Evidence from genomic data. Firstly, I'd like to present my research here in USP. I'm in the last year or last month of my PhD, and I'm part of the Mantelato's lab. And together we try to understand about the patterns and process of genetic diversity of decapod crustaceans in Brazilian marine ecoregions. You can see. In Brazil, we have 8 marine ecoregions. You try to understand if they work as a barrier for these crustaceans and the hotspots of genetic diversity too. So we selected 13 decapod crustaceans as a model to try to understand this process, patterns in process. So since my beginning here, when I start my PhD, me and Professor Mantelato already know that we wanted to establish an international partnership. So when the opportunity of the cap sprint appears, we decided to choose one of these species to understand more about the phylogeographic patterns. So we choose the University of Florida International University, so supervision of Professor Heather Bracken-Grissom and Dr. Pedro Perez. This university is located in Miami, Florida, and here's some pictures of the lab, of the building there. It's called Biscayne Bay Campus, and I spent my time there since July of this year. The lab there is called Crustomics, Crustacean Genomics is the Systematics Lab. We chose this place not only for the expertise of both professionals here, but they have a big collection there that I could analyze it and take samples that I needed to do this work. Also, Professor Heather, she's a partner of the lab, so we can use this for good. So there I could learn, understand, prepare and analyze my genomic libraries, and I had the opportunity to participate of some classes there with deep sea crustaceans, because we don't have this kind of species here in the collection. So I could enjoy it and we enjoyed the opportunity to do this class there. Also, I participated in so many events in the university in this time. I visited museums and I participated in some academic competitions there. This is a little famous. There's a lot. So, talking about my work that I did, like I said before, this is the shrimp that I chose. We call it Pneus brasiliensis or pink shrimp. It has a long distribution in the Eastern Atlantic here. And he's used a lot in gastronomy, so we catch for eating this shrimp here in Brazil, especially a lot. And the information about this valuable information about this species or this kind of group of shrimps is that they have a extended larval life cycle. So this is important to understand my results, and I'm gonna talk a little about it. There is a lot of soft marine barriers all over the world, but here in this region of the study, we're gonna talk more about the Amazon Orinoco Pool. That's the mix with the Orinoco River and the Amazon River here, and they change the salinity of this region. And this kind of barrier can create difficulties in predicting inter- and interspecific diversifications. And using genomic data in this case is crucial for understanding the diversification of marine species. So with this information, our aim was to investigate the phylogeographic patterns of these species and test the influence of this barrier on its interspecific diversification. And we hypothesized that this Amazon Orinoco plume reduced the connectivity and resulting to genetic groups. For this, we use two data sets, one using the gene CL1 with 40 individuals, and we also use the data of DD-REDSEC with 60 individuals using the reference genome of a closer species, the same genus, and use this data to obtain SNPs. So with this kind of data, we have samples of over its distribution, but because of some missing data in SNPs dataset, we need to remove some of them. We did analysis of relationships between individuals, genetic diversity, structure population, and structure genetic, and demographic history too. So the first result here you can see, we did a cladogram of maximum likelihood, and we can see two different groups. We can characterize. It's not working too well. Sorry. But we can see two groups here. These groups occurs in South, here in Brazil, and this group with the one sample here, we can see in the North, like represented there. So, but although they have two separate groups, we can see the genetic distance between North and South populations is only 2.32. So for this genus, there is not enough for now to separate two species, and morphological they don't have changes too. But we need more analysis to have sure. But knowing this, we move forward with populations analysis, and we analyze and map it. We mapped the genetic diversity using 3 different indexes, and we can see in all of them that we have more genetic diversity in North populations. About the genetic structuring, we did 3 analyses using the 2 data sets, and you can see in all of them that this analysis separate these groups in 2. And we named them of North population and South population. So after separating these groups, we can understand more about demographic history. So we did analysis of migration, and we can see the population of South, they have a migration to the North, but the opposite don't occur. And this this has a high bootstrap value or high support because it's the max. If you can see here, 1 in the middle, this is the max bootstrap value here. So there's no migration of populations from north to south. How about the effective population size? We can see they have expansion here in post-glacial maximum, and then in recent times they have a little decline. We cannot say that there's a higher decline there. It's not too much, but we can observe these details in both populations. But you can notice that in the south, this decline is more accentuated. Sorry. With these results, for now, we can conclude that the highly genetic diversity found in North may be due because there we found more effective population size, and maybe the decline observed in the last graph that I showed may influence these populations. The barrier reduces the gene flow among these populations, in like we hypothesize they separate in two. Also, the Amazon Orinoco Club barrier has a role in diversification species. So, not just in my work that I present here, but we can see this kind of patterns in a lot of invertebrate, marine invertebrate, and other crustaceans too. And the population decline that I'm just talking about, it can increase the strength of genetic drift and can help the diversifications in marine environmental. The next steps of this work is gonna do more investigation about the demographic history and do some models, and also try to understand more about the environmental adaptation and the impact of the fishing. Like I said, we consume these shrimps a lot, so maybe this decline could be influence of the recent fishing, and understand the current and future scenarios on their climate change. But for doing this last step, we need to separate a data set and use only locus on their selection. So I'd like to thank you, my supervisor here, Professor Fernando, and my supervisor in the United States, Professor Heather, for the opportunity, the program here, and the CapSprint for giving me a chance to know more about this culture and do my research there. The funding agencies that help me with the costs of everything, and also my friends here and the friends that I did there for all the happy moments together in discussing about science and personal life. Thank you so much for your attention, and I'm open to any questions.
Thank you very much. And your SNP analyses, could we go back to some of those results?
Which one is specific?
This one, the average it's very clean, it's very structured. I have a question about your process of analysis for this.
Mm-hmm.
Did you inform the pipeline of your populations for each sample, or did you just input the samples with no assignment to population?
In admixture analysis, you're talking about or both?
Admixture.
Admixture. Yeah, I inferred 4 populations, because we try to test the ecoregions first. In Brazil we have 5 there, so 4 by so I tried 4 populations, and they made 2.
So they were assigned.
Yes. But the test, this other analysis, we don't put the number of the max population we want. I like that because you can put geographic coordinates. So he only do this analysis of extrapolations. Thank you.
I have a question. I work with botany as you do. And I know that calling sequence is much used in animal phylogenies, for example. But I know that what is the length of the calling sequence? The length?
Between 500 and 600.
500, 600.
Base.
Why is it so different? Because it's very short.
Yeah, we use a lot in genetic populations because they have a variation, a good variation for trying to see some difference in individuals. But I'm agree that if you use more genes, you can infer. Yes. Thank you.
Well, now the last lecture. We have an international lecture now. It will be conducted by Dr. Linnea Hess from the Departament of Biology, University of Hamburg, Germany. And she will talk about image technologies in biological studies. It's more or less. Okay. And I'd like to thank very much Linnea for your presence here because it's not planned in her schedule, I don't know, and she's giving a course together with Professor Fernando Nogueira, that's here, and some students of the course is here as well. So thank you for your presence. And finally, we will have some plants in this symposium, because too much animals. Enough.
So it's a great pleasure to be here. Everybody thanks me for being here, but I'm so thankful because for me this is paradise and I don't want to go back, but unfortunately I'm a professor in Hamburg, so I have to go back. They need me there, but I'm having the time of my life here, and I thank everyone, especially also the course. You guys are showing me around and I get to see so many cool things, and now I get to talk about what I do, so this is just heaven. So, so I'm a botanist by training, but I work in the functional morphology, biomechanics, and biomimetics field. And I do a lot of imaging, which is all about visualizing and analyzing functionality in plants. And so what is biomimetics? Not many people know this field. It's where we try to learn from living systems for technological systems, and in my case, plants. So, for example, we look into biological fiber-reinforced composites, like plants or monocots in this case, and we want to learn from them in terms of design principles, fiber connectivity, fiber-reinforced composite manufacturing. Or what we also do in our work, we're with Fabian, Noah. I brought two PhD students with me because they have to see this place as well. So Noah works with the fiber-reinforced composites, and Fabian works with plant movements. So how can we learn from passive hydraulically driven plant movements measurements for movements in architecture, for example, hingeless systems, which are new and energy autonomous in their movements. Or something where we look into the repair of plant structures, how they can actually repair themselves. That's something very ubiquitous to all organisms. And we can learn from that in terms of damage management and self-repair and technology. So it's actually a pretty cool field to work in. And what we typically do is we study the relationship between outer shapes, inner hierarchical structuring or materials, and then functionality. And here again, like form can be an outer geometrical shape. Structure can be on multiple levels of hierarchy, from tissue elements to cells, cell walls, or molecules. So we have to study all these, or at least take a look into these hierarchical levels. And then somehow form and structure perform specific functions that we're interested in, and we really want to understand in a multifunctional organism a specific function. So that is not that easy to do. So form and hierarchical structuring and material are rather static, two-dimensional, three-dimensional to analyze, whereas functionality is very dynamic. Something happens over time, and multiple things happen over time, and you want to understand that one function. And we have a difficulty with existing methods to actually study functionality, and what we're doing in our work group is to try to optimize specific imaging techniques to better visualize functionality in biological material systems. So, for example, many people, it's become very popular is computed tomography. We know this from maybe some of you have been in a CT and broken an ankle or whatever. So you might have your own experience with it, but it's become very popular to also image plants. And this is a dragon tree, it's a monocut. It has vascular bundles and parenchyma tissue. But what we see is we don't really see anything if we look into a very fresh, aquaceous plant material. That's because water dominates the absorption coefficient of our tissues. But if we take away the water and dry the plant, we suddenly very beautifully see, for example, the vascular bundles. Or we can even go image on a single cell or cell wall level. So using computed tomography, we can image very large plant samples down to the nanometer scale. So we can cover multiple levels of structural hierarchy using this technique. However, there's some downsides, of course, and that is tissue contrast. So if you have plant material, we can just simply take away the water. This is critical point drying, and suddenly we see different structures within the plant, but it's not a good contrast. It's basically a contrast between cell wall and air. And then what we're currently trying to do is find contrast agents which are tissue selective. And well, it's not working too well right now, but this is what we call ProHands. It's contrast agents for humans in MR imaging, or MRI. That's also a medical imaging technique. And it does give small contrast in computed tomography as well, but it goes into anything, so it's not very tissue selective, and I'm really interested in simply just visualizing the vascular tissue, for example, and it did work that well. Imaron is also a medical contrast agent for computed tomography in medicine. We can use it in plants, and this is a sample where we let the plant work for us, so we gave it into the water of the plant, and it just sucked it up on its own. And there is this kind of tissue selectivity within the vascular bundles, but then we have a leakage of the contrast agent into the peripheral tissues of the cortex parenchyma. And this is a very popular contrast agent. It's it's tungsten-based, it's PTA. It's basically used since 2013 by numerous researchers globally. But we can see here in this, all of these images are on the same window level, so they basically have the same amount or the same region of pixels visualized. We can see that we have PTA as contrast agent in any cell wall, so it's also not tissue selective. It's popular, but we can't really say that I just want to see the vascular tissue or the pollen tube or whatever you want to contrast. However, the contrast is based on this absorption coefficient, and what really plays a role is the atomic number Z, and it differs between these different agents, and it increases simply the contrast between air or water and tissue. So suddenly we actually get a contrast, although these tissues are not dry, they're wet, whereas this one is dry, just for you to understand what happens. So what I work a lot with is MRI, because here we can contrast tissues using just the way the plant is designed. We actually use water for contrasting. This is a volume analysis of this branching region in the dragon tree, and we can see individual vascular bundles, highly contrasted to the surrounding parenchyma tissue. And if you look very closely, there's this really bright line surrounding the vascular tissues, and that is the meristematic tissue. So we have a very decent tissue contrast using a completely different technique. And if we compare CT, we cannot really look into living material in plants because of the, at least monocots, which have a lot of water in them. It's really difficult to image living plants using CT, but it is very easy to image living plant tissue using MRI. And just direct comparison, here the light microscopy image and here an MR image, we can see that the cortex and the parenchymal tissues are very dark. Hopefully the class of today knows why now. And these bright dots are the vascular bundles. They're very bright because there's liquid water in them. And then we have this very bright line where we see meristematic tissue. And this I like a lot, because if you see meristematic tissue, you can see where development is taking place, where the plant starts to grow. And since we can use this non-invasive method, it's based on magnetization and not ionizing radiation or something, we can actually watch a healthy living plant develop over time. But I'm going to get to that in a bit. What I'm going to talk about now is one of our projects and why this is so important to have these imaging techniques. So this is the project Noah works on, the one with the curly hair. Here we want to understand how we can improve fiber-reinforced design and technology. Here, for example, we have different materials. It's all black, it's carbon-based, so carbon fiber-reinforced material. It's not very good to recycle, and the idea is to understand how plants grow if they have a higher load-bearing capacity using their fiber-reinforced design, and then helping us to design very high load-bearing structures using biologically based fiber-reinforced material, which is much better recyclable, and maybe one day we can actually go into biodegradable as well. So the benefit of fiber-reinforced design, and we see that in all animals have that too, is that it's a very lightweight design. It has a high strength and stiffness while still being able to damp, which is important for plants. If you imagine them swaying in the wind, they can damp this due to their fiber-reinforced design. The challenges in technology, however, are the fiber connectivity. So if fibers are not connected, that is the region where we can have rupture and where things actually break apart. The fabrication of technical fiber-reinforced components is still very difficult to perform. That makes these components very, cost-intensive. And it's really difficult to take the material back into a production phase and post it production adapted. Whereas plants, they can respond over their entire life to changing environmental conditions. And then, of course, as I mentioned, I really want to get to that recyclability of fiber-reinforced technical products. So plants are wonderful inspirations because we find all types of geometrical shapes, branching patterns, and interior fiber-reinforced design. And monocotyledons, and many of these images are from 2018 when I was already in Brazil, so I keep showing them around the world. So these are all monocotyledons. Also, we find all different shapes, sizes, and designs, and they have a very pronounced fiber-reinforced material structuring on multiple levels of hierarchy. If we look into the dragon tree and take a cross-section through the stem, we can see that these little yellow dots are individual vascular bundles embedded in this reddish, liquidy cortex. So the vascular bundles are rather stiff, and then we have this viscoelastic, what we call it, parenchyma tissue. The individual vascular bundles are close collateral. We have the sclerenchyma fibers, so very lignified fibrous cells, and then we have the phloem and xylem for conduction, of course. And these vascular bundles run like fibers through the entire plant body, so we have a fibrous, reinforced structuring also on the tissue level. And if we study these monocots, we can study the macroscopic level, find fiber-reinforced design on a tissue level, fiber-reinforced on a cellular level, on an ultrastructural level of the cell wall, and then we can even go down into the biochemical level where we really have cellulose fibers embedded into the lignin matrix. So it's great models to study fiber-reinforced design on multiple levels of hierarchy. So what I always ask myself, seeing these plants is, how can they carry my load or these branches, and how do they grow and develop? And how can we learn from these plants? And we see self-supporting plants, we see, epiphytes, we see those that are able to climb with their roots, branch angle climbers. We have twining species like the Dioscoracea or the yams. So there's a lot of different growth forms and behaviors we see in the monocots that we can learn from and understand. And what I want to understand is, how do they design? How does the fiber-reinforced arrangement within these plants, and can we learn from that in terms of compliancy and motility, or rigidity, or the load-adapted arrangement of these fibers? And how can we learn from them in terms of fiber connectivity across complex three-dimensional nodal elements where we have attachment of laterals like branches, leaves, fruits, and so on, or rhizomes with root attachments? And then how can we learn from them in terms of growth and development, in terms of fabrication and technology? So what we did was use this really weird stick, which is non-magnetic, that allows us to place an entire living plant with roots and leaves attached to it into an MRI device. And then we studied the plant in a not loaded and then in a mechanically loaded condition where we applied a force of about 150 newtons to the branch. And using MRI allows us to actually study the deformation in 3D. So here we see the geometry, and red is the loaded condition, and we see the branch being pulled out and downward. But the cool thing is, I can look at the fibrous tissues being deformed. So we can study, here red again, how individual vascular bundles are being pulled out and in a downward direction. Why is this of interest? Well, we can quantify these deformations, so we have numbers to this visual data, but I just want to break it down. We were able to quantify that the vascular bundles in the branch stem attachment joint are being pulled away from the main stem in the upper region of the branch stem attachment and pushed towards the main stem in the lower region of the branch stem attachment while the branch is being pulled or bent downward. So we have compressive stresses down here and tensile stresses up there. We can feel these stresses if we bend ourselves, so we can feel something stretch and compress, right? This is something that happens within the branch stem attachment as well, and this is very typical. So this is a beam theory where we have a beam, and if we bend it downward, we will find tensile stresses in the upper region of the beam and compressive stresses in the lower region of the beam, and a branch is nothing else but a rather complex beam. But what's interesting is fibrous tissues like your fibers, like your shoelaces, they're not good if you compress them. They just buckle and fail. Now, that's basically what happens to vascular bundles, but they're massively lignified. So they have a compressive material that is good in compressive stresses within them. So instead of just buckling and failing, or this little fiddle, they actually have this sort of compressive resistance, but then they massively buckle outward and break. So, so what's interesting now is, if I go back again to this slide, regions where tensile stresses are dominant, we see that the vascular bundles are placed in direction of these pulling stresses. So they're adapted to tensile stress because vascular bundles are good if we pull on them. But in regions where compressive stresses occur, we have this curvature of the vascular tissue, and now the entire vascular bundle is pushed into the surrounding viscoelastic parenchyma tissue, which compensates these compressive stresses. This is important because if the vascular bundle would buckle and fail, there would be a massive injury which has the potential of being lethal to the plant, and this doesn't occur anymore. So what we're currently doing is working on methods to better visualize these deformation patterns using digital volume correlation technology, and that is something that Fabian and Noah, my PhD students, are working on very well right now, where we can quantify deformations a bit more straightforward and faster and reliant, so we can repeat these experiments on multiple monocuts. But how is this load adapted design developed over time? And we can image plants growing over half a year using MRI. It's non-invasive, it doesn't have an effect on the growth, speed of the growth. So what we see here are longitudinal sections basically through the branch stem attachment 4 days after I decapitated the plant. So if you cut off the apex, it developed branches, and then you can see these branches grow over the time frame of 180 days, and this is always the exact same location within the branch stem attachment over half a year. And if we draw a line through the center of this very young bud meristem, we basically draw the neutral plane through the biological beam, and suddenly we see in regions where tensile stresses are dominant, we have this load-adapted, tensile-resistant arrangement of vascular tissues, and below this line we have the compressive adaptation. We can do this in 3D because we have the 3D data set, so we can also study how this entire branch is actually attached to the main stem, which is really interesting in monocuts because they have different meristems they grow with. And what you can imagine this growth process being, it's very comparable to extrusion 3D printing. So basically, the plant prints its branch layer by layer onto the outer periphery of the main stem. So you can easily simulate this three-dimensional growth using a 3D printer. But we want to better understand how the plant knows or senses the mechanical forces in the environment. So Noah is working on diffusion tensor imaging, where we study molecules moving due to diffusive processes through the plant body, and we want to see if mechanical stimuli change diffusive processes, so we can detect changes in water diffusion within the plant locally, but also globally by studying sap flow dynamics through the entire plant body. So we're using MRI also to study water dynamics in plants, basically, that are linked to mechanical stimuli. But we compare many different species to also see, how do these monocuts differ in their arrangement of vascular bundles, and we can do this using multiple techniques. And what we also do is, I'm going to show you on these selected species how their mechanics change, and if this might be linked to the arrangement of vascular tissues. So we have the youngs, which are the blue ones. Flagellaria, which is a climbing grass, which climbs with leaf tendrils. Youngs climbs by stem twining, so there has to be different mechanical adaptations of the stem. And then we have Maranta, which is just cross over the soil. And here we have the length of the stem, and here we have the modulus of elasticity, which is the higher it is, the stiffer the stem. And we can see that very young, so if the stem is very long, it's very young. So young stages in Flagellaria are rather soft, and then it gets stiffer the older it gets. While the stem twiners of the youngs, they both, more or less maintain a specific stiffness, and then, of course, a plant that doesn't really have to stand upright is very soft. So that's just what you can kind of imagine with your mind, but we can also measure this. But what is really interesting is how do these plants that do not have the capability to grow by means of secondary thickening growth, how can they actually change their mechanical properties? And they do that by changing their cell wall properties. So they change the thickness, the amount of material, and possibly, and this is what we're looking at right now, also the orientation of cellulose fibers and their direction within the cell wall. And we can see very young stem regions of flagellaria, we see no lignification. Then a region where the plant kind of hangs in the surrounding vegetation, where the stiffness kind of stays the same. And that is where the vascular bundles already show lignification. And then we have this densification of the tissues with further also parenchymal tissue getting involved in the lignification process. And that actually gets the plants to become very stiff like bamboo. They're very closely related to bamboo as well. And they're that stiff that we can't cut them with a razor blade anymore. So it's really stiff, dense material. And the question is, can we get this, adaptation also into material systems? So we have a fiber or low adaptive fiber reinforced design that we're looking at. We have connectivity across nodes that we're looking at, and we have developmental processes that we are interested in. In order to bring this knowledge from plants into the manufacturing technique, and this is my colleague Larissa Born from the ITFT in Stuttgart, Germany, and we can see in white glass fibers, and in black we see carbon fibers. And the carbon fibers are arranged in a way inspired by the dragon tree. And what we're basically doing here is we're braiding this fiber-reinforced sock, which is then filled with concrete, and it's used, or was used, as a load-bearing pillar in architecture. So we're testing different material design to develop lightweight building. But again, this is carbon and concrete, so it's not very nicely to recycle. So currently we're working more on recyclability aspects. So I'd like to very briefly also talk about kinematics in plants. This is what Fabian, the not curly German dude, works on. So here we're interested in plant movements and how they inspire us for moving technological systems. And this is a plant called Hamamelis, witch hazel in English, and it has these fruits. And when they dry out, these fruits open by cracking open, and then inside of these fruits we have what we call an endocarp. It's a very woody structure, and it basically pushes on a seed, like your fingers push on a watermelon seed, and it flinges the seed out. You can imagine it flying like a football would fly. It has the self-rotating attitude, and it has quite a flight distance. So you can see it can fly up to 72 meters in a very quick, explosive outburst. And we wanted to know where exactly this force is being applied on the seed, and we could identify using MRI this little pressure region, which is why we know that it flinches out like a seed would, like watermelon seed would flinch between our fingers. And currently we're looking into this bomb here. This is a explosive dynamite tree fruit. It's about the size of an orange, and also upon drying out or desiccation, we see these little carpels crack apart, and in a massive explosive outburst, which is a big bang, we see these shrapnels of a bomb flying through space with a speed of 240 kilometers per hour. So it's a very massive explosion. And this force is somehow kept within the material system, and we're really interested in understanding how a woody lignified fruit material can store such energy, and we can use this to store energy in moving technological systems. And this is what Fabian does a lot, so we try to understand how do these carpel moves, where do they fracture, why do they fracture, how the tissues within this carpel, so this is a section through here, and we see there's an endocarp which has an outer endocarp layer and an inner endocarp layer, and these layers differ in their properties. We're currently looking into cell wall components, so the material, the orientation of the individual fibers. We're studying using synchrotron technologies, the swelling and shrinking behavior of single cell walls to really understand how this plant performs this outburst. So we have a colleague who uses his hands to cut out single cells from this plant, and then we place these single cell layers or cells into a nano-CT, and we use different relative humidities and image the same cell wall, for example, with 0 relative humidity and then with 90% of relative humidity. This is cross-section, this is longitudinal section, and then this is an overlay of both situations, and we can see there's a swelling occurring. We can calculate the amount of tissue swelling, so it swells by 17.7% in this sample here, and this cellular swelling and shrinking behavior eventually leads to this explosive process. And we want to, for example, use this with a colleague in Darmstadt, Germany, to further optimize these self-actuating, facade-shading systems in architecture. So we save energy. These facade-shading systems respond to either the environmental relative humidity, temperature, light. So depending on the material you use and place into your facade shading systems, you can use different stimuli for these facade shading systems to move automatically, passively, without our, active participation. And this is I call it the M cube of biomimetic research. So I'm a botanist by training, so I look into the plants and study the plant. We develop the methods to better understand functionality in plants. You can use these methods also for animals, of course. And then we work very closely with material experts or engineers and try to transfer these biological principles and functionalities using further developing material system and manufacturing systems into technological products. And during this biomimetic research, we do not only learn for technology, it always also goes backwards, so we learn for nature as well, which is the fun part. As botanists, of course, we really understand the biological role models much deeper. And with this, I have to thank, of course, my entire team and the people I collaborate with, the people that give me money, my wonderful students here, Noah and Fabian, are with me right now. And I don't know, I'm having the time of my life with you. I have to really thank Faye for getting all of this set up. She's doing a fantastic job and showing us around. And thank you for your attention, of course, and having me here.
Time for a few questions.
Thank you, Leah. Wow, a lot of things. When we work with plant anatomy, for example, so I give classes in plant anatomy. One of the most challenging things is to put in a student the 3D aspect of the morphology of a stem or a leaf. And do you also work with this approach in your lab, or the didactic approach to classes, for example, and material to give classes to undergraduate and graduate students?
Yes, so I remember myself being a student, and plant anatomy just blew my mind, and that's when I became a botanist. I actually started studying biology out of different reasons. So, and I always was annoyed by the fact that everything was visualized in 2D, and with my class, I now, of course, give them the 3D overview of the stuff that I make, what Fernanda does, what other researchers globally do in 3D, just to show them the three-dimensional arrangement of tissues and cells, and it's just images, especially in these multidisciplinary fields that I work with, are very important communication techniques. You show someone, like a mechanical engineer has a different language than I do, and if I want to tell them, this is the important part, I just show it in a picture. We develop three-dimensional models. You can, everything you saw in 3D on here, or basically 3D on here, we can print out and carry as individual models into the classes and actually have people touch them. So we're definitely trying to bring this this knowledge of plant anatomy, which is so fun and cool, into classes for people to really touch and experience. Yeah, that was the question, right?
I have a question. That's fine. Okay, can shop.
Yeah, you go shop.
My question is, first of all, congratulations. I do love applied science. It's like amazing. My question might be a little bit technical, and I want your advice. This MRI technology that you applied to get all those details about the vascular system in the plant can be applied on a dead material like fossils, or it needs water?
Wonderful. Wonderful question. So she just asked if we can apply MRI also to dead fossils, for example. There are many people working on that. My colleague, where Noah is working at right now, they actually imaged a mummy head. And so you have different atoms. I taught it in the class today. So you have different atoms in the material, and they have to have an uneven number of protons and or neutrons. And for example, you can use fluoride or different salts, and go specific on that if your material doesn't have any hydrogen. But if you have organic compounds and they have a hydrogen linked to it, you can definitely do it. So you can play around with other chemical signals. We're detecting hydrogen atoms, and that's why we see water that well. But you can also detect other things. So yeah.
Perfect. So I might try to find you somewhere to know more about this technology.
I also have a card. You can always, I can give you my card later on, and then we can also stay connected.
This is interesting for paleontology. So
Yeah. Well, thank you for the great question. Yeah.
Yeah. I would like if you could clarify a little bit about the projects. To make it clear. Okay, you work with basic science, okay, but you have a joint project with applied science with people from engineering. Is that correct? So it's established by the university, this kind of joint project.
Very good question. So in Germany, biomimetic research is getting more and more popular. And the typical way, if you really want to get into a good product, it has to be a big collaborative project. And we have two different ways to do that. The idea starts in the nerds at the university. So the scientists sit together and we're like, okay, we can actually realize it. So you get together as big teams with architects, engineers, and biologists, and sometimes the engineers ask us for advice, or we discover something really fun and cool and ask the engineers, do you think this could be something cool for you? But sometimes we also have industry asking us. So there's been automotive industries asking us, or airplane construction, and like building companies that just ask for advice. But in order to get such a project really out on the market, you need all of them to be involved. In industry, you want the results to be there in a very short period of time to have a very good outcome. But if you really want to understand functional principles in nature, you need more fundamental research. So typically, I'm more in the fundamental part because I'm also very interested in the method development, because anatomy is so fun for me. So a lot of my projects are also just university, but then when we have detected functional principles in more detail, then we go into the second phase and ask colleagues in industry to support us in these projects. Yeah.
And the first step is what? The basic science, or the product that could be get with this kind of science?
Either way, actually. So we have what we call biomimetic push and biomimetic pull, or bottom up, where we scientists are like, oh look, this can does something really fun, it explodes with such an energy, we need to look at it. That is definitely a biomimetic bottom-up process where we start with a biological role model. But then we have industry coming or colleagues coming and saying, okay, listen, we really need to develop a self-repairing material. Can you help us with some organisms? And that is what we call a biomimetic top-down. Yeah, and I work with both projects. Right now we have more bottom-up, but I just started at a new university and I have to build up my lab, and then once you're more stabilized again, I started in Hamburg this year, so I still need a bit of time, but then we're planning to have projects also with architects and someone from the United States, and I have one industry project right now where we're building this little wood-based pavilion for the German Bahn, and that was also more a top-down project where this guy approached me, for example. But you can have all of them, yeah, and typically it's good to have all of them. Well, thank you guys so much.
Thank you, Linnea. We're going to give another, Linnea? Oh, sorry. You're going to give another lecture in this afternoon, but it's not the same thing, another one. In the Café Consciência lectures.
It's similar but not identical.
Not identical,
Yeah.
so people, it's not the same, so again, come back again to Linnea. Thank you, Linnea, very much. Linnea will be here in Brazil till Friday, in Ribeirão Preto, till Friday.
20th, we're going to Brasilia, and then we're going to the Amazon, and we're going back 20th of this December.
Okay,
Yeah.
okay, thank you very much. Thank you, Fernanda, and guys. Well, now we have the lunch break, and after the lunch break will be the poster awards, and after the closing ceremony, okay? But I'm very happy until now, because we could see the high level of lectures that we have in this set of presentations, both from the lecturers, the professors that are advisors here, from Linnea, and also from students. So we have a high level of presentations, and I'm, as a coordinator, and Tomás as a vice coordinator, we are very happy with that. Okay, so we come back 2 o'clock afternoon, so have a good lunch, and see you soon, okay? Well, now we are going to final part of our symposium. So in our schedule, there are 2 activities. First one is the poster awards, and after the closing of the event, okay? So let's go. It's so funny. I made it myself. Okay, some numbers first. We have 34 posters evaluated with 48 voters that are much more than we are expecting yesterday, and 216 votes, because each posters with 5 evaluations. When you remember, you can remember. Okay, the prize, okay? It'll be 150 reais in vouchers on Amazon.com, right? And the prize will be equal to everybody, right? And we had no idea the numbers of the voters, the number of the posters that will be presented. So we considered winners the people with more than 10 votes. Okay, everybody with more than 10 votes in the poster is a winner, right? Any winners with no more delays. In the 8th position, Karina da Silva Alencar with 11 votes. Guys, this is not fake, okay? Because it's my signature, so you can get your prizes, okay? Congrats. Now the 7th place. Jessica Scaliorigalo. Jessica here? No.
Em inglês, velho.
She asked us to submit a Cyrus prize or call her because they go out to
But she's not here, so...
no problem.
Okay. So Jessica is from São Carlos. Huh? That's why she's not here. Malu Araújo Almeida. Congrats, Malu.
Thank you.
It's not fake. Is there some photos of your leaders? No. What you got? Yeah, good idea. One with all the winners, right? Better, yeah. Congrats, Malu.
Thank you.
The 5th place, Ana Laura Silva Paiva. Congrats, Ana Laura. The 4th, Guilherme Borges Pimenta.
Opa, cadê Guilherme?
Então, melhor. Acho que ele não veio.
No? Sorry.
Sorry.
Congrats, Guilherme. Okay. 2 guys here. And now the podium with the 3rd place, 2nd, and the first one, the 3rd place, Paulo Roberto Furiri. AKA Paulão. Congrats, Paulo. Very good. Now, the 2nd place is o Raí Coelho, Brasil de Souza. Where is Raí? No? Congrats, Raí. It's here. Okay. And the 1st place with, I think, 24. Yes, 24 votes. Otávio Gomes Victoria. AKA 13. Not fake. Guys, thank you very much. Congrats for the winners. Of course, we have votes to every poster, okay? Some of them with 9 votes, some of them with 8 votes, but the participation of you yesterday was amazing. We, Milton and Thomas, we were very happy with the participation of the students yesterday, and it's just for fun, just to informal talk, and I think it was good because we've seen the students really discussing the project in English. So to me it was an amazing night as a professor, as a coordinator, as an advisor. Okay, thank you very much. And now, one note that was interesting yesterday is many of the voters were graduate students from our course, people from the first period in our course of undergraduate students, maybe 6 or 7 of them, some of them with from the 3rd year of our course. So it was amazing to see these people participating, taking part in this kind of event, because we can call these people to get part of the comparative biology. Many of us were students from our graduate course. Otávio, Vinícius, Igor, Martin, wow, okay. Anna, that is now Fozolki. A lot of people that took part in the undergraduate course.
And yesterday you have the participation of the other students, entomology.
Yeah, for the Pós-Graduate Program in Entomology as well. Yes. And now we are going to close our ceremony, and I would like to ask you to talk in Portuguese, okay? If I may, okay? May I? Okay, thank you. É estranho, tem coisa que a gente só consegue falar na nossa língua mãe, né? Então vamos lá. Primeiro de tudo, eu queria agradecer todo mundo, eu como coordenador, Tomás como vice-coordenador. Né? Então esse foi um evento muito importante para mim, do ponto de vista pessoal. Por quê? A gente fez esse é o quarto evento, e o terceiro foi online, então a gente não teve contato com os alunos. Eu queria muito fazer em presença, como é que é em português mesmo? Presencialmente. Presencialmente, né? Então eu queria muito fazer um presencialmente. Só que esse ano eu tive um problema muito sério com a minha esposa, não sei se alguns de vocês sabem, né? Ela teve um derrame e eu fiquei muitos meses assim por conta da minha esposa. Então eu queria ter feito isso muito antes, né? Mas não consegui, ficou coisas da vida. Mas eu não queria sair da coordenação sem ter feito um evento desse presencial com vocês. E os dois eventos que foram feitos, né, o tanto de autoavaliação, que a gente fez em agosto, quanto esse agora também, para mim foram importantes porque foi o fechamento de um ciclo da minha participação. Né, o Tomás agora ele vai assumir, né, como eu falei, daqui a pouco eu vou falar do Tomás também, vou passar a palavra do Tomás também, mas para mim foi importante porque completou um ciclo. Né, e agora a gente tem algumas tarefas para a gente fazer, a gente tem coisas para fazer, mas eu acho que o programa, isso foi mostrado nesses 2 dias, né, ele está muito bem, né, a gente tem um corpo de discentes que está muito engajado, né, e isso acho que vai também aparecer muito no encontro de Biologia Comparada do ano que vem. Eu tenho certeza que assim, vai ter muita gente participando da organização, né, e eu acho que essas coisas que fazem diferença, quando a gente tem os alunos engajados, os professores engajados também, a gente consegue trabalhar como se fosse um time. Né? Então as pessoas sabem os objetivos. Né? Alguém falou, acho que foi o Otávio que falou para mim, falou assim, olha, agora a gente sente que a gente tem objetivos, né? E que a gente tem um rumo, que a gente tem um caminho. Né? Então a gente pode batalhar para esse caminho. Tá? Quer falar algumas coisas antes, Tomás? Depois você passa para mim ou não? Não? Você sabe. Então eu queria agradecer vocês, tá? Agradecer muito mesmo. Vou continuar com o professor do curso, né? O que eu puder ajudar também no curso eu vou ajudar, quando solicitado. E queria agradecer aos pós-docs que vieram aqui, aos alunos, aos professores que participaram. E eu queria também agradecer para uma pessoa especial, que eu trabalhei com ela durante 8 anos agora, né? E que está ali atrás toda preocupadinha com o vídeo, né? Está de vermelho lá. Vem cá, velho, um pouquinho.
No fair. Do you remember yesterday? I'd like to First of all, I'd like to say thanks God, because if you was not here, I'm not here. And excuse for everybody, because the pandemic was terrible for me. Someone knows. To say thanks for Wafa when I come back here after pandemic. He make a video for me. I will not forget.
I love you.
see the compare. It was necessary that I call the students, but she, when I come back, did it for me. I say thanks. Excuse me, because everybody knows me, knows that I'd like to be kind for everybody, but pandemia made me feel so strange person. I don't know what's happening with me. Mas, enfim, estamos de volta, no fair, the new coordinator, and the coordinator could contact with my help. And like he told you yesterday, we have a lot of work to do, and everybody has a capacity to work together. We're going together and help someone, each other help the other. And I can, if you need some things, I'm here to help you. I love everybody. I love my work. They know that. I'm here to help you, to do my best, not the best of the world, but my best I do.
Isso aqui, vou pedir para os alunos que estavam aqui, depois você lê.
I'm a 65-year-old, gente.
Então, gente, muito obrigado também por vocês terem colocado essas mensagens para a Vera, a gente fez isso. Enquanto você estava lá, a gente estava fazendo, eu estava te enganando. Tá, então agradeço todo mundo também, e um presentinho. Presente também, já de Natal. Isso. Aí depois você vai ver que o presente tem um significado. Se você quiser ver agora também, a gente pode até explicar.
I forgot. I'd like to say excuse me for my voice because when I come back, I don't know if you remember. I put the chair in in the door for nothing. Come back like me. It was so terrible. I don't like this.
Só para eu entender, esse aqui, para quem é dos Andes, né, esse daqui é um tecido do Equador, e ele tem as cores de uma divindade que é a Mãe Terra, que é a Pachamama. E eu acho que combina muito com você, né? Você realmente...
Uma vez um coordenador falou para mim assim, nossa, mas por que você trata bem os alunos? Por que eu trato bem os alunos? Sabe o que eu disse para ele? Primeiramente, porque eu sou assim, eu não estou forçando isso. E segundo, vocês passam aqui 2 ou 4 anos e vão embora. E muitos vêm de fora, muitos não são daqui. E isso fica assim uma marca na gente da necessidade de acolhimento a vocês. E, coitado, o John também falava para mim assim, que você é mãezona. Eu falo, não, eu não consigo driblar os Jantos, eu não sou mãezona. Mas veja, se eu tenho possibilidade de fazer o meu melhor, por que eu não vou fazer? Está em nós, é igual o nosso trabalho, igual o Tomás falou, vamos fazer junto. Vamos nos ajudar, a gente erra, fala para o Milton me ajuda. Às vezes a cabeça não funciona, né? Essa semana também ele falou para mim, para a gente pensar juntos. Então é assim, gente, é um ajudando o outro, né? A gente está aqui de passagem, todo mundo está aqui de passagem, né? Eu queria entrar muito na USP, mas eu não consegui fazer faculdade, então entrei depois de aposentada para trabalhar na USP, porque era um sonho meu. Mas enquanto eu estiver aqui, já falei para o Milton, para o outro coordenador e já falo para o futuro. Eu me senti que eu estou fazendo parte do jogo, eu estarei aqui. A hora que eu vir que já não tenho mais nada a fazer, eu vou embora. Então, enquanto eu estiver aqui, é o meu melhor. O meu melhor para vocês. E nós todos juntos somos uma equipe.
Isso aí, velho. Mas eu não vou sumir, não. Ainda vou ficar por aqui. Gente, então... Então é isso, muito obrigado a todos pela presença de vocês. Então, declaro encerrado o nosso simpósio em Comparative Biology. Desejo toda a sorte para a nova coordenação, professor Tomás, professora Fernanda. Eu acho que o programa está em muito boas mãos. Eu acho que a gente tem o caminho certo para trilhar e vamos na fé que a gente consegue os nossos objetivos. Muito obrigado, pessoal. É isso aí.